Hello, everyone. Thank you for joining us for today's webinar, Navigating Regulatory Challenges for Cell and Gene Products in 2024 and Beyond. I'm Leslie Bebko, your host from Cell and Gene and Life Science Connect. With me today from Cardinal Health Regulatory Services, I have Doctors Ash Carr, Director of Regulatory Affairs and Product Development, Yin Lai, Principal Scientist, Chemistry, Manufacturing and Controls, and Matthew Varkey, Supervisor, Chemistry, Manufacturing and Controls, Regulatory Operations Strategic Solutions. Over the next 45 minutes, they'll be discussing the complex regulatory landscape of cell and gene therapies. After their presentation, we'll be holding a live Q&A session. Feel free to submit your questions as we go along. We'll get to as many of those as possible during the event. Any of those we can't get to, we'll follow up on afterwards. If you run into any issues, please try refreshing your browser first. And if you need additional support, submit a question via the Q&A. Finally, this webinar will be available for on-demand viewing, and you'll receive a link via email. And with all of that said, I'm going to turn it over to Ash to get us started. Ash?
Thank you so much, Leslie. Me and my colleagues at Cardinal Health Regulatory Sciences are glad that you've joined today. Now, this slide presents our agenda for today's presentation. I will be first speaking to the recent and upcoming regulatory developments that are applicable to cell and gene therapy products, following which my colleague, Dr. Lai, will be speaking to the importance of control strategies for ancillary materials for the cell and gene therapy products. And then lastly, Dr. Varkey will be providing insights about the regulatory considerations for development of companion diagnostics. Now, I would like to start my presentation by highlighting the importance of year 2024, as this year will revolutionize the regulatory landscape for advanced therapies. At a recent press conference, Dr. Peter Marks, the director of CBER, stated that 2024 is expected to be a breakthrough year for gene therapy approvals due to the new initiatives being implemented to promote the development of these products. Now, the Congress and FDA have taken this task seriously and released multiple directives, not only to implement the new initiatives, but also improve the efficiency of existing regulations to implement the experience gained so far for these regulations. Now, in my portion of the presentation, first I'll be speaking to FDA's recent guidance on modernizing the accelerated approval pathway. As you may be aware that the accelerated approval pathway allows for a product to be approved under conditional terms based on the use of a surrogate or clinically intermediate endpoint instead of an endpoint that is solely based on clinical outcomes. Now, that is important if certain conditions are met. Now, it is eventually required for the applicant to prove efficacy in a confirmatory trial prior to receiving full approval of the product. Thereafter, I will be speaking to upcoming regulatory initiatives that include issuance of platform technology designation and advanced manufacturing technologies designation. Following those, I'll share an overview of the regulatory and clinical considerations for human genome editing products, to which FDA has openly stated that it will consider accelerated approval pathway for bringing such products to the patients in dire need of new therapies for the rare genetic disorders. As you may be aware that the accelerated approval pathway is going to be the norm for gene therapies, especially with the newly released guidance on human genome editing products. Before we get to the new regulations that are intended to improve the efficiency of accelerated approval pathway, we first have to understand the inefficiencies of the regulatory procedure itself, as it's been in existence since 1992. In the year 2022, the Office of Inspection General initiated a review of the performance of accelerated approval pathway. Now, this was initiated following FDA's controversial approval of aducanumab, a drug intended to treat Alzheimer's disease. Now, the results of this research are published and available in the public domain. Now, in this graph, the red bars represent the drug applications approved under accelerated approval pathway and had incomplete confirmatory trials past their initial projected completion dates at the time of approval. Now, since the accelerated approval pathway began in 1992, the drug applications have steadily increased, with overall 278 approvals by CDER from its inception to the end of 2021. As you can see, the last few bars on the right are higher than the ones on the left. Now, 1/4 of the approvals occurred in the last 2 years preceding the year this research paper was published, that is, in the years 2020 and 2021. Now, of all the overall 278 drug approvals, 104 had incomplete confirmatory trials. For the remainder 139 applications with completed trials, it took an average 4 years from when FDA granted the accelerated approval to when FDA deemed their trials to be completed. Now, the key takeaway from this research is that more than one-third of accelerated approvals with incomplete confirmatory trials were past their trial's original planned completion dates, including 4 approvals that were in fact more than 5 years past those due dates. Now, in December of 2022, Congress passed the Food and Drug Omnibus Reform Act, also known as the FDORA, which was enacted in 2023. Among other things, this act gave FDA additional authority to require that the confirmatory trials be underway at the time of approval. It also offered an additional enforcement mechanism if the sponsors do not complete these studies with due diligence. Now, under this act, if a product is granted accelerated approval, and if FDA determines that no post-approval study is required, FDA must publish a rationale on its website explaining why such a confirmatory study is not required. If, however, a post-approval confirmatory study is required, FDA must specify the conditions for the required study. These conditions may include enrollment targets, study protocol, and milestones, including the target date of study completion. Additionally, the sponsors must report the progress of any required study, including progress of any conditions specified by FDA. Now, prior to enactment of FDORA, the progress reports on these studies were only required annually. However, under FDORA, sponsors must report the progress of any required study no later than 180 days following approval and at least every 180 days thereafter until the study is completed or terminated. Now, this change enables FDA to initiate enforcement actions in a timely fashion for a failure to conduct required post-approval study with due diligence, including a failure to meet any required conditions that have been specified by the FDA. Now, FDORA 2022 further describes the procedures that FDA and sponsors should refer to when withdrawing the accelerated approval. Under this act, FDA first provides a notice to the sponsor, including an explanation of the proposed withdrawal of the accelerated approval. Following this, the sponsor has the opportunity to submit a written appeal and request for a meeting with FDA's commissioner or designee. Thereafter, if it is determined that the withdrawal procedure should proceed, the notice will get published by the FDA in Federal Register for public comment. FDA will then provide the publication of summary of the public comments received and the Secretary's response to such comments on FDA's website. Now, this act also provides sponsor and FDA to have an opportunity to hold the FDA Advisory Committee meeting if Advisory Committee has previously not advised on withdrawal of this product. Now, if upon completion of this whole regulatory procedure, FDA still considers that withdrawal of the accelerated approval should occur, the notice of FDA withdrawal of the accelerated approval is published on FDA's website, including withdrawal of a product from appropriate FDA databases such as the Orange Book. Now, this slide here provides a case study of the first ever withdrawal of an accelerated approval by FDA following enactment of FDORA. The product Pepaxto received accelerated approval in February of 2021 for use in combination with dexamethasone for the treatment of adults with relapsed or refractory multiple myeloma. In September of 2022, an advisory committee meeting was convened to discuss the results of the confirmatory trial that was underway, following which FDA in August of 2023 provided notice of withdrawal to the applicant since the confirmatory trial had failed to verify the clinical benefit and Pepaxto was not considered to be safe or effective anymore. Now, within a month following that, applicants submitted a written appeal and opportunity to meet with the commissioner's designee. However, following additional discussions, FDA made a final determination that Pepaxto still needed to be withdrawn from the market and was removed from the market in February of 2024. Now moving on to the next topic, which is the designation of platform technologies. The Prevent Pandemics Act has included a new designation program known as a designation for platform technologies. These are the technologies that have the potential to increase efficiencies in the drug development. A platform technology is eligible for designation by FDA if these three conditions are met. The first condition is that technology has incorporated in or is utilized by an approved drug or biologic. Secondly, if the preliminary evidence demonstrate that the technology has the potential to be used in more than one drug without an adverse effect on the quality, manufacturing, or safety. And then the third condition, which should be met as well, is that the technology has a reasonable likelihood to bring significant efficiencies to the drug development or manufacturing process. Now, FDA will provide a response to such a request in 60 days. The designated platform technologies may be eligible for expedited development or review actions, including early interactions between the FDA and the sponsor, timely and interactive advice from the FDA, and even prior inspections. Now, after the designation has been granted, and at any time if these criteria are no longer met, FDA has the authority to revoke this designation. We are aware that FDA is actively working on a draft guidance document to provide additional guidance to the industry on how this designation program will fully become functional. Now let's talk about the Advanced Manufacturing Technology, or in short, the AMT designation. FDA guidance on AMT designation was released in December of 2023. This is yet another initiative by FDA to improve efficiencies of development of advanced therapy products. Under this program, a method of manufacturing or a combination of manufacturing methods is eligible for designation as an AMT if such a technology incorporates a novel technology or uses an established technique or technology in a novel way that will substantially either enhance the drug quality or improve the manufacturing process for a drug and maintain drug quality by either reducing the development time or by increasing or maintaining the supply of a drug that is either on the drug shortage list or is a drug that is life-supporting, life-sustaining, or is of critical importance to providing healthcare. Now, prior to requesting such a designation, FDA strongly recommends that applicants engage the CDER's Emerging Technology Team, or CBER's Advanced Technology Team, prior to requesting this designation. The contents of the AMT designation request are provided in the guidance document, and briefly, they include the description of the manufacturing process and technology, including the rationale why the technology fulfills the criteria for the designation, and any prior engagements with relevant teams within the FDA regarding this designation. The application can also cross-reference other relevant applications and data that are relevant to this designation request. Now, the application is submitted over an email to the FDA, and FDA is expected to review the applications in 180 days. The key incentives of the AMT designation include early interactions with the FDA regarding development of the AMT, expedited development and review of the applications, including FDA to provide timely advice, interactive communication, and even involvement of FDA senior management. When a designated AMT is used across multiple drugs, the knowledge and familiarity that FDA gains during assessment of the first application should streamline the assessment of subsequent applications that use the same designated AMT. Now, this is my last slide. As you probably are aware, that FDA has released two guidance documents in January of 2024, one for the cell therapy products and another one for the genome editing products. Now, in the interest of time, I'll be only focusing on the human genome editing guidance in this presentation. And this slide does provide a brief overview of the guidance document. Now, in general, FDA strongly recommends that the sponsor of human genome editing products communicate with the OTP, or Office of Therapeutic Products in CBER, early in product development stage, that is, before the submission of an IND, to discuss the product-specific considerations for transitioning these products to the clinical phase of product development. These interactions typically include the Type B pre-IND meeting and the Interact meetings. The Interact meeting can be used earlier in development, even prior to the pre-IND meeting, to discuss issues such as non-clinical development or manufacturing processes so that the sponsors can obtain non-binding regulatory advice. The Human Genome Editing Guidance from FDA provides details on clinical considerations for clinical trials with such products. The aim of the clinical trial is to assess the safety of the gene therapy product itself and the additional risk associated with gene editing. When applying for an IND, the overall study design, assessment of AEs, and subject follow-up plans should be well described in the IND. Clinical trial design should appropriately define the patient population, an efficient and safe approach to product administration, including database dosing and dosing schedule, and the treatment plan. It should also provide the details on adequate safety monitoring and appropriate safety and efficacy endpoints. Now, a sponsor of such a clinical trial should select the study population carefully. In general, the patients with advanced disease are at higher risk of having more safety issues with the study population and will be willing to accept the risks associated with the use of an investigational product. However, in order to understand the safety first, it may be more appropriate for the patients with less advanced or more moderate disease be included in the first-in-human trial. Now, the next topic is the dose and dosing schedule, which should be derived from the available non-clinical data and the data or experience gathered from the clinical use of similar products. Now, typically, the first-in-human trials are conducted in a stepwise fashion to start with a lower dose and often lesser number of patients. Staggering intervals should be of sufficient duration to detect acute and subacute adverse events. It should also take into account the expected duration of activity of the human genome editing product. As I mentioned previously, the safety assessments are critical for gene therapy products, and a safety follow-up of up to 15 years could be required. Now, the clinical trials include monitoring for any off-target editing and an adequate assessment of outcomes of off-target editing and unintended consequences of on-target editing as anticipated from the non-clinical studies. Now, with regards to the endpoints, as I mentioned previously, FDA is supportive of the use of accelerated approval for such products and encourages sponsors to discuss potential eligibility of accelerated approval, including proposed surrogate endpoints or intermediate clinical endpoints early in the development program. Now, with this, I will conclude my presentation and pass on the floor to Dr. Lai for next portion of her presentation.
Thank you, Ash, for the introduction. Good morning. I'm Yin Lai, and today I would like to discuss the material aspect of cell and gene therapy development. In December last year, FDA actually published a draft guidance on potentially assurance plan, which include multiple factors. One of that is the control strategy. Within the control strategy, there is an element for material control, which I'm going to discuss today. As we all know that for cell and gene therapy product, it cannot be sterilized. Therefore, the quality and safety of the ancillary material used during the manufacturing process for cell and gene therapy product is of very important. Before I discuss more on the material aspect, I would like to discuss on the current guidance that are available on material and terminology used. In US, ancillary material is used, whereas in EU, raw material is used. Per USP 1043, ancillary material is defined as substances or material that are used in manufacturing or processing, and they are not intended to be present in the final product. And this material can include multiple categories, for example, plasma or human serum-derived, biological extracts, buffer, cytokine, growth factor, and so on. For raw material, European Pharmacopoeia 5.2.12 have a definition for raw material, which is any other substances used for manufacturing or extracting the active substance, but from which this active substance is not directly derived, such as reagent, culture media, fetal calf serum, buffer, and so on. In addition to the European Pharmacopoeia and USP, ISO also published Technical Standard 20399, which also provide a definition for ancillary material. In addition to that, it also provides two levels for ancillary material. Level 1 is correspondent to the product level, in which ancillary material does not contain any material from animal or human source. The level 2 corresponds to the production level, in which it defined the ancillary material is produced without the use of any material from an animal or human source. So these are the current general guidelines, I would say, and terminology used for ancillary material. For the purpose of this presentation, I'm going to use ancillary material. In addition to what I have discussed earlier, ancillary material also can be categorized based on source and types. For example, single component versus multiple components or more complex ancillary material, compendia versus non-compendia ancillary material, and different categories: serum, growth factor, media, antibody, protein, enzyme, and so on. There seems to be some inconsistency right now in the quality grade in the ancillary material produced in the industry right now. For example, the quality grade can be research grade or RUO or research use only grade, laboratory grade, xeno-free, GMP, CGMP, or GMP source grade, high HQ or a high quality grade, clinical grade, and so on. So the vendor are using different terms for ancillary material. However, one quality grade, the same quality grade may be defined differently by vendor A versus vendor B. Therefore, sponsor needs to communicate and to know what is the definition of the quality grade for the ancillary material you plan to acquire. In addition to that, ISO Technical Standard 20399 also provide definition for animal-derived component-free ADCF and chemical defined material. USP 1043 also provide a risk classification on ancillary material. There are actually 4 tiers. First tier corresponding to the lowest risk, whereas Tier 4 corresponds to the highest risk. For Tier 1, it corresponds to low risk, highly qualified material, such as approved drugs, biologics, and clear device, container closure, or transfer system. Whereas Tier 2 corresponds to low risk, well-characterized material, which is mainly produced under CGMP and non-animal derived material, such as density gradient material, media, and sterile process buffer. Tier 3 corresponds to moderate risk that require relatively higher level of qualification relative to Tier 1 and Tier 3 recombinant growth factor, cytokine, or novel polymer, scaffold, and hydrogel. Whereas Tier 4 corresponds to the highest risk, which require extensive qualification from the vendor and also used under CGMP, or complex ancillary material and animal-derived ancillary material will be in this category. For example, fetal bovine serum, animal-derived polymer, scaffold, and hydrogel. In addition to the risk classification, USP 1043 also provide general guidance on the factors impacting material risk assessment. To control the material is to assessment should be performed at the early stage of development. There are multiple factors that affect that sponsor needs to consider. when performing the risk assessment for your material. The first is source and complexity. Sponsor needs to know the source of this material or origin of this material, where it's coming from, and the more complex the material is, the higher risk. The material derived from animal or human will have higher risk compared to the material which is chemically defined. And also, since ancillary material is not intended to be present in the final product, therefore the amount of ancillary material used in your manufacturing process should be considered. The higher the amount of material used in your manufacturing process, then the risk will be higher. And material criticality. Whether this ancillary material is critical to your manufacturing process. Material which is critical will have higher risk. And the next one will be removal step and proximity. These two come hand in hand. If the ancillary material is introduced at the upstream of your manufacturing process, which is further away from your final product, then the risk will be lower versus the ancillary material that is introduced at the step that is closer to the final product. And of course, the final factor would be the phase of development. At the earlier stage, less data support is needed versus relatively to the later stage of development. There are also multiple methods of analysis tools that are being used for risk assessment. One of the popular ones is the FMEA. There is a failure mode effect analysis, which can be used for risk assessment. In addition to that, there are other tools, for example, fault tree analysis. After the sponsor has performed the risk assessment for the ancillary material, the next step will be to perform the material qualification. USP 1043 also provides general guidance of material qualification. Qualification is actually an exercise, a process in which the sponsor will acquire the data to establish the source, purity, identity, and safety, and also, more importantly, the suitability of your ancillary material to your specific manufacturing process. So the level of qualification should reflect the level of risk of your ancillary material for your manufacturing process. Therefore, it is important to perform the risk assessment first before the material qualification. But USP 1043, there are a few factors that sponsors should take into consideration. The first one is identification. So sponsors should list out all the ancillary material which are being used in the manufacturing process, the amount, the intended use, and the step in which you introduce this material into your manufacturing process. The second would be the selection and suitability, to look at the assessment of microbiological, purity, identity, biological activity, and also potential infectious diseases or advantageous agent of your ancillary material. And if your material is of animal derived, to look at the country of origin and also the TSE, BSE evaluation. In terms of characterization, a sponsor should perform characterization to look at the quality attribute of your material in terms of identity, purity, functionality, and safety testing, for example, microbiological or viral safety testing. And the level of testing should be based on the risk assessment, which should be performed before the material qualification. And in terms of vendor, due diligence for the vendor should be performed, and preferably on-site audit for the manufacturing facility. And it is important to establish a quality agreement with your vendor, which also should include the advance notification of any change to the ancillary material. And finally, sponsors should have procedure in place to segregate, inspect, and test the incoming ancillary material, and to have additional testing if needed in terms of stability and additional characterization. Also, procedure should be in place to manage the out of specification material. the verification testing should be performed if needed to verify the testing that are performed by the vendor of your ancillary material. In addition to that, the temperature and condition of storage of your ancillary material is important, and proper leveling and tracking of your ancillary material should be taken into consideration when a sponsor performs the material qualification. After the risk assessment and material qualification have been performed by the vendor, the next step would be to have established the material management and control strategy. There are multiple factors that sponsors should consider, and a lot of these factors I have discussed earlier under the material qualification, but there are a few points I want to add. In terms of vendor and material qualification, if that ancillary material is critical to your manufacturing process, it is recommended to have the dual source ancillary material and implement this new source in your development early, so that the sponsor will have time to evaluate the ancillary material from two different sources. And also, if the ancillary material, critical ancillary material, is single source, then a sponsor is recommended to have a supply agreement with your vendor to ensure continuous supply of your ancillary material and to have additional stock of this ancillary material. For critical material attribute, this should be identified by the vendor, especially for your critical ancillary material. This will help the material vendor control the quality and safety of your material. With identification of your critical material attribute, then the sponsor can establish the acceptance criteria for the ancillary material. So both the critical material attribute and the specification will help the vendor to control the quality of safety of your incoming ancillary material. And the next one would be traceability. The sponsor should pay attention not just the quality, but also the origin. Where is the material coming from, and have a traceability of and tracking of your ancillary material. In that sense, that sponsor would know what additional testing needs to be performed and what specific characteristic needs to be controlled to ensure the quality and safety of this ancillary material. And I want to stress again that a quality agreement is important, which should include notification of change that should be established with your vendor. In addition to that, there are a few other considerations, for example, in terms of regulatory and quality documentation support. So when selecting your ancillary material, the vendor, so it's preferable to have the vendor with a master file in place. This will help the registration or application process to be smoother, because there are multiple regions in this world that accept master file for ancillary material. For example, US, Canada, Japan, and China. For the other region like EU that does not have this system, then the vendor can request early on the regulatory support documentation or package with the vendor. So the expectation of the sponsor should align with your vendor. So make sure that this documentation is provided. And also a certificate of analysis, certificate of compliance, a certificate of origin, and TSE, BSE statement, if applicable, for animal-derived material. So this quality documentation are important. Additionally, sponsors should look at secondary animal-derived component if it is used in your ancillary material, and human-derived material that is outside of United States. And also additional advantageous agent testing may be required. For example, if the sponsor is using insect cell line, the SF9, then sponsor should test for spiroplasma and also different type of insect viruses of the world. For example, in terms of testing requirement, and some material may not be acceptable in other regions. in certain region of the world. In addition to that, a sponsor finally has to look at the interaction between the ancillary material with your product and also your ancillary material with your packaging, and also the performance of your ancillary material in the context that you are using that in your manufacturing process. I think currently there needs to be a harmonization and standardization in terms of quality and documentation in ancillary material. And there are multiple organizations that have initiated the initiative in doing that. One of them is a standard coordinating body, which has an initiative of defining a different aspect of certificate of analysis for ancillary material. So this concludes my part of presentation. And next, my colleague Matthew is going to talk about companion diagnostic. Matthew?
Thank you, Ian. Hello, everyone. I'm Matthew Workey. In this part of the presentation, I'll be discussing regulatory considerations for companion diagnostics and regulatory processes that can be leveraged to facilitate successful submissions. So companion diagnostics are regulated in vitro diagnostics devices that provide information essential for safe and effective use of a corresponding drug or biological product. They are used to identify patients who are most likely to benefit from a particular therapeutic product, to identify patients likely to be at increased risk for serious side effects as a result of the treatment with a particular therapeutic, and also to monitor response to treatment with a particular therapeutic product in order to basically adjust treatment for improved safety or effectiveness. Companion diagnostics are regulated by FDA based on the risk of the test, more specifically based on the risk to the patient if the diagnostic results are inaccurate. When they are used for drug approvals, companion diagnostics need to comply with regulations. Their performance is critical and it goes on the drug label. The drug label will refer to an approved or a clear diagnostic test, whereas the diagnostic label will refer to the corresponding drug. So the drug and diagnostic submissions are intrinsically related. So it's extremely critical to plan early. This schematic basically represents an ideal scenario of the co-development of a companion diagnostic along with its corresponding therapeutic, where a single analytically validated test is used and perfected until the registration trial. And then a final lockdown version of this test is used to select patients. And finally, a market-ready companion diagnostic application is submitted for same-day approval along with the NDA or a BLA. But unfortunately for us, this is not an ideal world. So the most common scenario involves playing catch-up with respect to the companion diagnostic development. And so this necessitates bridging studies. So there are several reasons for this. I mean, just to give you a few examples. These may include cases where a variety of laboratory-developed tests are used to enroll patients. And so there are no test negatives available to validate the companion diagnostic. Sometimes early phase data is used for drug registration, and then there are resulting specimen availability issues. In some cases, it could be that the diagnostic partner company or the pharma company, which is a sponsor, makes mid-trial test changes or changes to the cutoff, leading to challenges in the clinical validation of the companion diagnostic. Or some other times, it could be just that the patients were selected for a specific gene variant, but at the conclusion of the study, it was found that patients with many other variants were enrolled due to the use of different types of tests. But unfortunately, what may happen is the companion diagnostic is not intended to be used with that particular, with the therapeutic, and is not designed to detect any of these variants. So as you can see in all these cases, it will be extremely challenging to obtain same-day approval of the drug and diagnostic without extensive assessments. So the best way to deal with such contingencies is to plan early and engage with CDRH earlier. So this all begins with defining the biomarker and the technology that will be used, because that will actually define the patient population. So you basically define the biomarker analyte, whether it's DNA or RNA, and the test specimen site, because eventually CDRH will need analytical validation for each claimed specimen site. In subsequent slides, I'll basically discuss in more detail about investigational device exemption, risk assessment, as well as bridging studies. So moving on, it's important to have a specimen acquisition plan to bank specimens from patients evaluated and request for adequate samples in order to complete analytical validation. Have a plan to obtain test negatives. Another great strategy is to have paired specimens, which means you could have blood and bone marrow. You could have liquid biopsy as well as tumor sites. Make sure to consider the impact of storage on the stability of specimens. If you have multiple sites, specify uniform methods for specimen handling and pre-analytical steps. When you get to the clinical validation, the assay should be fully specified and locked down. The instruments, reagents, and softwares used for the validation should be legally marketed. They cannot be research use grade. This holds true for the blood and biopsy collection devices too. Another important aspect is you need to have appropriate informed consent for the test validation. Moving on, it's important to plan submissions such that the reviews of both the drug and the corresponding companion diagnostic are basically, the reviews are aligned for same-day approval. If it's a class 3 device, you could make use of the modular PMA process, wherein CMC, software, and analytical validation all can be submitted as separate modules ahead of your clinical validation data. You can also consider use of human device exemption category if it's a rare disease and you intend to run under 8,000 tests per year. In terms of planning, you'll need to allow time for BIMO inspections and manufacturing. You'll need to get letters of cross-reference as well as master device files in case of confidentiality from your partners. You can use pre-submission meetings to obtain feedback, which is very timely and non-binding from CDRH. And it could be anything regulatory, be it study protocols, statistical analysis plans, or even regulatory strategy. I'd also basically like to call it attention to the Breakthrough Device Program. This is for devices that provide for more effective treatment or diagnosis of life-threatening or irreversibly debilitating diseases or conditions. And if granted designation, this is a great opportunity to have more frequent and faster interactions with the FDA, as well as have a prioritized review of your submission. So FDA regulates medical devices, including diagnostic tests based on the specific clinical use. Investigational device exemption, or IDE, enables the use of investigational tests for patient diagnosis and treatment. An IDE will be needed irrespective of the phase of the trials or the number of patients involved. And keep in mind, this applies only to US sites testing foreign patients and foreign sites testing US citizens. It does not apply to foreign sites testing foreign citizens. Also, ID requirements are based on risk to the patients. All clinical evaluation of devices must have an approved ID before study initiation, unless they're exempt devices. So exempt devices are those devices which whose results are not reported to the physician for treatment decisions. Whereas when you look at non-exempt devices, they fall into basically two categories. They could be either significant risk or non-significant risk. And both have similar requirements. The main difference being significant risk studies need a submission of a full ID application to FDA prior and approval, obviously, before it's used in a trial. Whereas a non-significant risk test is assumed to have an automatically approved ID, provided the sponsor follows the ID requirements. So how is the risk category of the device determined? There are quite a number of ways for that. Basically, your IRB can make this determination. And if you get a decision from your IRB, then you don't need to check with FDA. Although there are caveats to this, there are few rare cases when FDA has looked at the risk and then overturned the IRB's decision. You could also submit a study risk determination request to CDRH through a pre-submission, or alternatively as a question in the original IND. The agency basically looks at risk and determines risks based on 4 criteria. They look at whether the patients are foregoing more effective treatments. They look at whether the subjects are exposed to excessive safety risk compared to the standard of care or the control arm of the therapy. They also look at whether the patients or subjects who are not supposed to be enrolled, would they have more adverse events if they were to be enrolled on the therapy? Another critical question is, would there be invasive sampling just for the sole purpose of testing the biomarker, thus exposing the patients to unnecessary risks? So along with giving you this information about risks and investigative device exemption, I also want to point out that typically only about 5 to 10% of companion diagnostic submissions need full ID submissions. Now, coming to bridging studies, it could be a nightmare, but if handled well, it could go smoothly. Now, bridging studies are needed when the test used in the clinical trial is not the version that is intended for marketing. A bridging study typically is a statistical plan to assess the concordance between the results of the clinical trial assay and the companion diagnostics. And it basically assesses the impact of any discordance on the original efficacy conclusions. The goal basically is to confirm that the companion diagnostic will support the selection of the same population and hence the efficacy of the therapeutic. So for this purpose, all positive and negative specimens need to be retested. It would be great if specimens are annotated for test performance and factors that affect therapeutics so that it can be demonstrated that there are no biases due to any missing samples. It is also important to have informed consent that allows for retesting. So in summary, the biomarker and technology that will be used needs to be identified because this will decide the target patient population. The clinical trial assay strategy needs to be defined because this will affect timing and ability to get specimens. Also, you need to ensure adequate specimen collection for validation studies, and you need to coordinate submission timing. Overall, the key is to a successful submission is basically to plan early and engage with CDRs early. Thank you for all your attention. Now I'll pass it on to Leslie for the poll question as well as the subsequent Q&A.
All right, thanks, Matthew, and our other presenters as well. Again, if you haven't submitted your questions, please do so now. We do have a question for you before we head over to those. So, are you interested in connecting with an expert from Cardinal Health Regulatory Sciences? Yes or no? I'm going to pop that up for you. If you could please answer that for us. And then we are going to head to the Q&A. So if you please just take a moment, yes or no, whether or not you would like one of our experts to connect with you. And then we are going to head over to our Q&A. So Let me look into what we have here. And just a reminder, everyone will be receiving a link automatically to this presentation on demand. I know we had a couple of questions come in as to whether or not the presentation would be available. Yes, it will. You'll receive a link to that automatically as soon as it is. So let's go here. Yin, I believe this one is for you. How can sponsors better prepare for marketing application when master files are not available for ancillary materials?
Yep. Thank you for the question. I think, as I mentioned earlier, sponsors try to select a sponsor vendor or ancillary material with master file. However, that is not can not always will be true, will be available. So in that case, when a master file is not available, then a sponsor should talk to the vendor early on to see what documentation can be provided to the sponsor. For example, there is a regulatory support document package some vendor actually offer, and if and different type of quality documentation, for example, as I mentioned, certificate of origin, certificate of compliance, TSE, BSE for animal-derived product. So that should be provided. So the expectation of the sponsor should align with the expectation of the vendor. So this I recommend the sponsor to discuss with the vendor early on during the ancillary material or vendor selection process, so that the marketing application will be process will be smoother for the sponsor. I hope that answers your question. Thank you.
Thanks, Yin. All right, moving on. Matthew, I believe this one is for you. Do all these considerations hold good for therapeutic agnostic companion diagnostics?
Yes, more or less. The only difference between agnostic diagnostic applications would be you may not necessarily have same-day approval. But all of them hold good for one-to-one as well as agnostic ones.
All right, thank you. Will all the gene therapy products be eligible for accredited approval pathway?
I think that's essentially not correct. I think what FDA is really trying to say here is that many of the genome editing products may be eligible for accelerated approval. I think now we really have to go to the basics here because, as you know, accelerated approval pathway is for serious conditions and the conditions where there is high unmet need. So I think those are the preliminary requirements. And following that, there also needs to be a surrogate endpoint or an intermediate clinical endpoint that is reasonably likely to predict the clinical outcome. So I think those are the basic criteria that do need to be met. And of course, in some of the conferences which we have attended and some of the speeches we've listened to from FDA, that they will be supporting initiatives that are involving advanced technologies, and also for the patients that are in need. For example, the patients with rare disorders, and also where oncology is still a very big area of interest. So I think that's a long-winded answer to my question, but I hope that answers it.
Thank you. Will the new regulations lead to lower number of approvals through the accredited approval pathway?
That's anybody's to guess, but I think there are so many programs that are in development for gene therapy products that we do anticipate the numbers to keep going higher. I think, but the new regulations do support that as well, that more approvals will be considered acceptable by FDA for accelerated approval. As I said, if the conditions for accelerated approval are met, and also for rare gene disorders where this will be of very importance. I think so. I would say I don't think the number is going to go any lower, but I think it's going to go higher over the years.
All right, thank you. Moving on, let's see. Could you please share your thoughts on if there are any regulatory requirement or expectation for biocompatibility assessment of ancillary materials?
Okay, yes. As I mentioned earlier in my presentation, so as a sponsor, your responsibility is to also evaluate the interaction between the ancillary material with your product and ancillary material with the packaging or container closure that you use. So if you have a whole step in your manufacturing process, so then evaluation need to be performed. But for the vendor, if you are referring this to the vendor that provide manufacture your ancillary material, then it is a responsibility for the vendor to evaluate the biocompatibility of the ancillary material in the container closure that is being used. So, however, you as a sponsor should acquire that information from your vendor. Currently, I don't see a clear-cut requirement for biocompatibility of the ancillary material in the guidance right now. Per the agency, sponsors should always use the highest quality of the ancillary material available at the current stage. So I would say for sponsor, you talk to your vendor to make sure that you get the information you need for your application to ensure also the safety and quality of the ancillary material you receive from your vendor.
Okay, and there's, I believe it's a follow-up to that, or maybe a clarification on that. With that, is there a regulatory or sponsor expectation for the ancillary materials supplier to perform assessments on biocompatibility, but then mentioning extractable and leachables? So I don't know if you have anything to add with that.
Yes. So biocompatibility, extractable, and leachable, that is a responsibility of your vendor which provide the material to you. But you as a sponsor should get that information from your vendor, and to ensure that you will get a quality product from your vendor. So I think, therefore, during the selection process, this discussion needs to be met with your.
All right, well, thank you so much. I think we are coming up on the hour. So if you had submitted a question, I definitely see one in there from Marianne and a few others, but if you'd submitted the question, we didn't have a chance to get to it, we will follow up with you afterwards. But to my presenters, thank you so much for joining us today. Great information and material. And to everyone joining us from the audience, we really appreciate your time today. Thank you so much. Hope to see you again soon, and enjoy the rest of your day.